Hello everyone, welcome on the platform of Unacademy. Let's crack CBSE Science. Good afternoon to each one of you. So today we are going to have our discussion on the question paper of 2015. Okay, so yesterday we have discussed the two question paper. One was from the question paper 2016, one was 2017, and today we are going to learn about the 2015 question paper. Okay, so before we start, I would like to give my introduction first, that I am Divya Gupta. I have done MSc in microbiology. I have around 3 years of teaching experience and I am completely passionate about teaching. That is what about my introduction. Let's talk about the Unacademy's two different subscription options. One is the Iconic and another is the Plus subscription. In the Iconic and in the Plus, education that you are going to get will be completely same. Okay, there is no at all difference. Only the difference is that उन दोनों के जो फीचर्स है उनके अंदर ही सिर्फ डिफरेंस है. तो बात करते हैं पहले आइकॉनिक सब्सक्रिप्शन की. तो इसमें मिलेगा आप लोगों को पर्सनल मेंटर, मीन्स यू कैन हैव डायरेक्ट वन टू वन इंटरेक्शन विथ योर मेंटर एंड यू कैन कम डायरेक्ट इन कॉन्टैक्ट विद द educator and you can clear all your doubts and the queries. Live doubt solving sessions will be there for you. Your parents can also get connected to us. Okay, they will be having the weekly report. Then you are going to have all the benefits of the Plus. Now let's talk about the Plus subscription. So in the Plus you are going to have daily live classes. Okay, then you will be provided the structural course. This structural course will help you to best prepare for your particular examinations. Then live tests and the quizzes will help for you. This will help you to analyze your performance, and then you will be having the unlimited access to all the courses, and the courses are available from class 6 to 12, and for all the competitive exam, that is more than 30 competitive exams courses are available on this single platform. So to have the access to all the free live classes, you have to make use of the code, that is DG007, so that you can have free access to all the live classes. Okay, got it? Now what we will get, that we have already seen. This is how the live classes looks. Then you are being prepared with the top educators, expertise at your home itself. This is the comprehensive syllabus. Yes, what you have to do, you have to download the Unacademy application. If you have not yet done, make an account over there and select the course for which you have downloaded the Unacademy application. Go through the free live classes. Okay, to have the access to the free live classes, make use of the code, that is DG007. Okay, and then click on the get subscription. For subscription, you are having two options. Before that, we are also available on the Telegram application. Download the Telegram app from your Google Play Store. Type this URL link and click on the join button. Okay, so in this Telegram group, you are going to have all the information regarding which educator is going to come live at what time, and each and every information, every update you are going to get over there. Then if you want to have Plus subscription, then select Plus. Iconic ka lena ho to select Iconic. So for 6 months, as you all know that you are having only 6 months of time for your class 12 preparations. So for 6 months in Plus subscription, the cost was 10,000, but now it is just costing 9,000. That is, you are going to have 10% discount by making use of the code, that is DG007. Okay, so my dear students, this 10% discount is all yours, and DG007 code is also all yours. Thing is that make use of it. And ये जो code है ना DG007, इस code को कोई भी use कर सकता है, चाहे वो student class 6 to 12 का हो, चाहे वो कोई भी competitive exam का student हो. This code can be used by any of the students. ऐसा नहीं है कि आप class 12 के biology student हो तो आप ही use कर सकते हो. It's not at all like that. Okay, for Iconic, for 6 months the cost was 17,000, but now it is just 15,300. That is, again you are going to have 10% discount. Okay, by making use of the code DG007. Is that clear? So that is what about the introduction. Now let's start with our question paper discussion of 2015. Okay, fine. So I think it's completely visible to you, right? The question paper is completely visible to you. Just write in the comment box, or should I little bit zoom in, zoom out? I suppose that it's clear. If not, then let me know in the comment box. I will try it out. Okay. So starting with the section A of 2015. Don't forget students, we are on the question paper of 2015. Okay. Now comes the section A, that is question from 1 to 5 will be of 1 mark each. Okay. So here comes the first question. Name the transcriptional active region of chromatin in न्यूक्लियस. न्यूक्लियस के अंदर जो ट्रांसक्रिप्शनली एक्टिव क्रोमेटिन का रीजन होता है उसको हम बोलते हैं यूक्रोमेटिन. ओके, विच इज ट्रांसक्रिप्शनली एक्टिव, या हम उसको नॉर्मल लैंग्वेज में बोले तो जहाँ पे ट्रांसक्रिप्शन अभी हो रहा है, ठीक है, उस रीजन को हम बोलते हैं यूक्रोमेटिन रीजन. नाउ कम्स द नेक्स्ट क्वेश्चन, दैट इज क्वेश्चन नंबर 2. A genetics interested in studying variation and pattern of inheritance in the living being prefer to choose the organism for experiment with short life. Provide a reason. ठीक है, अभी क्वेश्चन क्या बोल रहा है कि कोई भी जेनेटिक्स हो जिसको मतलब पैटर्न ऑफ इनहेरिटेंस या हमारी बॉडी हमारी लाइफ के बारे में पढ़ाई करनी हो, दैट इज पैटर्न ऑफ इनहेरिटेंस या वेरिएशन, तो दे ऑलवेज चूज अ ऑर्गेनिज्म विच इज हैविंग अ वेरी शॉर्ट लाइफ साइकिल. ठीक है, जिसका लाइफ स्पैन भी कम होता है और जिसकी लाइफ साइकिल भी बहुत छोटी होती है. ठीक है, ऐसा क्यों? बिकॉज मैनी जनरेशन कैन बी ऑब्टेन इन अ वेरी लेस पीरियड ऑफ टाइम. ठीक है, बहुत ही कम टाइम में बहुत सारे ऑर्गेनिजम्स को हम वी कैन हैव मैनी जनरेशन. ठीक है, तो जैसे इकोलाई है, तो इकोलाई का जनरेशन टाइम कितना है? जस्ट 20 मिनट्स. मतलब इन एवरी 20 मिनट्स इट डबल इट्स नंबर. ओके, तो अब डबल हो जाएगा नंबर तो यू विल हैव मोर पॉपुलेशन, एंड इफ यू विल हैविंग द मोर पॉपुलेशन then you can study more and more variations. Okay, so we can study the variations that have been existed. Okay, and select they can be selected faster. वरना अगर साइंटिस्ट जो है अगर वो हमारा ह्यूमन बींग को यूज़ करते हैं फॉर देयर एक्सपेरिमेंट, तो ह्यूमन बींग नीड्स एटलीस्ट 9 months ऑफ द टाइम टू गिव बर्थ टू द न्यू ऑर्गेनिज्म. ओके, एंड फिर वापस वो ऑर्गेनिज्म को मैच्योर होने के लिए वेट करो, फिर अगेन दैट ह्यूमन बींग विल टेक 9 months ऑफ टाइम. तो एक एक एक्सपेरिमेंट के लिए 9 months तक का वेट करना इट्स नॉट एट ऑल ईजी. नॉट एट ऑल ईजी मतलब बिल्कुल भी नहीं. ओके, so that's why the scientists use this smaller organism and the organisms which are having a very short lifetime. Okay, generation time बहुत कम हो. Next question, question number 3. What is biopriacy? Illegally, non-authorized, non-compensated use of bioresource by organization. So if any organization is there, I'm not talking about any of the particular organization, एक्स वाई जेड कोई भी ऑर्गेनाइजेशन हो, ठीक है, अगर वो बायो रिसोर्सेज को यूज़ कर रही है, बायो रिसोर्सेज मतलब इलीगली कोई पर्टिकुलर ऐसी जगह को यूज़ कर रही है जहाँ पे ऑलरेडी बड़े बड़े एनिमल्स रहते थे, छोटे छोटे एनिमल्स रहते थे. ओके, या दूसरा कोई बायो रिसोर्स, इट कैन बी एनी वाटर, इट कैन बी एनी लैंड. ओके, so when they are using the illegally that area or that place or that thing, we will call it as biopriacy. Fine. Now comes the question number 4. State a reason for the increased population of dark colored moth consists coinciding with the loss of lichens during the industrialization period in the England. ठीक है, तो पहले क्या था, इंग्लैंड के अंदर जब इंडस्ट्रियलाइजेशन नहीं था, इन दोज डेज देयर वर दी लाइट कलर्ड मॉथ. ठीक है, सारे के सारे लाइट कलर्ड मॉथ. बट बाद में क्या हो गया, इंडस्ट्रियलाइजेशन की वजह से बहुत सारा पॉल्यूशन लेवल बढ़ गया, एंड बिकॉज ऑफ दैट एवरीथिंग वॉज ब्लैक ब्लैक ब्लैक. सो मॉथ हैव ऑल्सो डिसाइडेड टू हैव देयर कलर टू द डार्क कलर ताकि वो लोग प्रीडेटर से बच सके. एक्चुअली दिस क्वेश्चन इज फ्रॉम योर चैप्टर ऑफ इवोल्यूशन एंड दैट हैज बीन स्किप्ड फॉर दिस ईयर. ओके, नाउ कम्स द नंबर 5. Indiscriminate diagnostic practices using X-ray etc. should be avoided. Give one reason. ठीक है, क्यों ऐसा बोला जाता, प्रैक्टिस है, we should avoid it. जब तक बहुत ज़्यादा ज़रूरी नहीं हो, we should not go through the X-ray because it is carcinogenic. Okay, it can cause the chromosomal aberrations or damage to the DNA. And I can say the normal cells can get converted into neoplastic. ठीक है, तो because X-rays they are the useful for our body, they are out of the range of our body. Okay, तो इस टाइप के रेज को हमारी बॉडी के अंदर आती है, so to the body. Okay, now comes the section B, that is the question 6 to 10 will be of 2 marks each. Okay, 1 mark questions were the 5 and 2 mark questions are again they are the 5. Okay, so my dear students, whenever you are writing the answer, okay, whether it is 1 mark, 2 mark, 3 mark, 4 mark or 5 mark, it doesn't matter. The thing is that how you are writing. 2 mark का question है तो you have to write the answer. It doesn't matter if the answer is so big, but if it asked in the 2 mark question, then you have to write it very very precisely. You cannot write the whole page for just the 2 marks. Now comes the question number 6. Differentiate between ZZ and XY type of sex determination mechanism. Okay, very good, very easy, very simple question. XY किसमें मिलता है हमें? In the human beings, and in the human beings XY represents the male. ZZ type का sex determination we use in the birds. Okay, and in the birds the ZZ is the male. Okay, मतलब क्या हुआ कि birds के अंदर होमोगैमेटिक कंडीशन किसमें मिलती है? We find in the male. While in the humans the homogametic conditions किसमें मिलती है? फीमेल में. Heterogametic conditions मेल में. Got it? XY और XX टाइप का सेक्स डिटर्मिनेशन किसमें मिलता है? In the human beings. XY that is heterogametic, it is male in humans, and XX that is homogametic, it is female in humans. While the ZZ and ZW type of sex determination, it is found in birds. In birds, ZZ that is the homogametic condition, it is the male, and ZW that is the heterogametic condition, that is the female. Okay, Dipanshu, hello, good afternoon Dipanshu. So let's move ahead. An infertile couple is advised to adopt test tube baby program. Step 2 principal procedures adopted for such technologies. Ma'am 2015 का पेपर है. Yes, this is the paper of 2015. Yesterday you were not there, but yesterday we have discussed about 2016 and 2017 paper, and today it's, मैम मुझे पता है, क्या पता है आपको? That this is 2015 paper. का आंसर I didn't got you. Okay, so why the infertile couples are advised to have the test tube baby program? Because IVF, that is in vitro fertilization, outside the body is almost, it is done in the similar condition as it happens inside the body. Okay, so later what happens, the egg, the, I can say the embryo that is formed, that it will be of 8 to 16 cell stage, that egg is then transferred into the female uterus. Yeah, ICSI, that is intracytoplasmic sperm injection. Here the sperms are getting injected. मैम 7th वाला दोबारा समझा दो. वही समझा रही हूँ दीपांशु, that is what I am explaining. Okay, when the couples they don't have their own child, okay, they cannot have their own child, so they are always advised to have the test tube baby, okay. And we have to write the two principal procedures, any of the two principal procedures, okay. तो एक था IVF, that is in vitro fertilization. दूसरा हम देख रहे हैं ICSI, intracytoplasmic sperm injection. Here the sperms are getting injected in the female body, okay, and it comes direct in contact with the ovum. Then ET, that is embryo transfer. इसके अंदर क्या होता है, द एम्ब्रियो जो हमने टेस्ट ट्यूब में एम्ब्रियो बनाया होता है, वो एम्ब्रियो को जब वो मैच्योर हो जाता है, दैट इज 8 to 16 सेल स्टेज में आ जाता है, बाद में उस एम्ब्रियो को उसको ट्रांसफर कर देते हैं इन द यूट्रस, ओके. ET, दैट इज एम्ब्रियो ट्रांसफर. इंग्लिश में समझ नहीं आ रहा, यस आई एम एक्सप्लेनिंग इन द हिंदी. ZIFT, दैट इज जाइगोट इंट्राफेलोपियन ट्रांसफर. इसके अंदर क्या होता है, जाइगोट को ट्रांसफर कर दिया जाता है, एम्ब्रियो में 8 to 16 सेल वाला सेल ट्रांसफर कर दिया जाएगा. जाइगोट में, जाइगोट जैसे ही बना, ओके, देन इट इज गेटिंग ट्रांसफर्ड. देन कम्स दी IUT, दैट इज इंट्रायूटेराइन इंसेमिनेशन, अर्ली एम्ब्रियो ट्रांसफर टू द यूट्रस, ठीक है. तो ये जो लास्ट के तीन है, दैट इज ET, ZIFT एंड IUT, ये तीनों जो है ना वो एक्चुअली एम्ब्रियो ट्रांसफर के ही स्टेजेस हैं, अलग अलग टेक्निक्स है. किस स्टेज में एम्ब्रियो को ट्रांसफर कर दिया जा रहा है बॉडी में, उसके हिसाब से ये तीन अलग अलग मेथड्स हैं, ठीक है. नाउ कम्स द क्वेश्चन नंबर 8. Many freshwater animals cannot survive in marine environment. Explain. Because फ्रेश वाटर के अंदर अमाउंट ऑफ सॉल्ट कंसनट्रेशन बहुत कम होगा, मैराइन के अंदर सॉल्ट का कंसनट्रेशन बहुत ज़्यादा होगा, ठीक है. तो ऑल्सो वी कैनॉट हैव मोर ऑफ दी सॉल्ट वाटर, ठीक है. हम बहुत सारा नमक वाला पानी हम भी नहीं पी सकते. ये तो चलो ठीक है, एनिमल्स है. अगर हमने भी बहुत सारा नमक वाला पानी पी लिया तो वी आर गोइंग टू फील लाइक फैटिगनेस और हमें चक्कर आने लगेंगे, बिकॉज बॉडी में सोडियम का लेवल बहुत ज़्यादा बढ़ जाएगा, ओके. हाई सॉल्ट कंसनट्रेशन आउटसाइड, इट क्रिएट्स हाइपोटोनिक सराउंडिंग. हाइपोटोनिक सराउंडिंग के टाइम पे क्या होता है, लॉस ऑफ वाटर फ्रॉम दी बॉडी. हमने देखा है ना कि ये एक जैसे मीडियम है, क्योंकि इसके अंदर सॉल्ट कंसनट्रेशन बहुत ज़्यादा है, एंड देयर इज़ अ सेल इसके अंदर सॉल्ट कंसनट्रेशन कम है. सो देयर विल बी अकर दी ऑस्मोसिस. यहाँ पे होगा ऑस्मोसिस. ऑस्मोसिस में क्या होता है, सेल एंड द सराउंडिंग वो हमेशा क्या करते हैं, एक दूसरे को बैलेंस करने की कोशिश करते हैं, ठीक है. तो यहाँ पे इस कंडीशन में क्या हो रहा है, हाइपरटोनिक सराउंडिंग क्रिएट हुआ है, मतलब बाहर वाले एनवायरनमेंट में सॉल्ट कंसंट्रेशन ज़्यादा है, इस वजह से सेल क्या करेगा, इट विल लॉस इट्स वाटर. सेल इज गोइंग टू लूज इट्स वाटर इन ऑर्डर टू मेंटेन द इक्विलिब्रियम. सेल क्या करेगा, अपना वाटर लॉस करेगा सो दैट इट कैन मेंटेन द इक्विलिब्रियम. दैट्स वाई द फ्रेश वाटर एनिमल्स कैन नॉट लिव इन द मराइन एनवायरनमेंट, बिकॉज जैसे ही फ्रेश वाटर एनिमल्स मराइन एनवायरनमेंट में आ जाएंगे तो उनकी बॉडी बिल्कुल सिकुड़ जाएगी, एकदम ख़त्म हो जाएगी, बिकॉज उनकी बॉडी में से सारा का सारा पानी जो है वो लॉस हो चुका होगा. नाउ कम्स द क्वेश्चन नंबर, इट्स दिस इज नॉट रिक्वायर्ड, ओके फाइन, लेट इट बी. Now comes the question number 9. Enumerate 4 objectives for improving the nutritional quality of different crops for the health benefit of human population by the process of biofortification. Okay, so how we can improve the quality, improving protein content, okay, oil content and quality को हम कर सकते हैं, vitamin content and micronutrient content. Now comes the question number 10. So again this question is a repeated question that was asked in the 2016 and also in 2018. थोड़ा सा changes करके the same question is there in the 2015 also. So now you can see that as much as more practice you will do for the old question paper, you can have more repeated number of questions and different form of the questions can also be seen by you. Okay, so up to here you have understood everything. You have to write the use of these organisms. First is LAB. LAB, that is lactic acid bacteria. It will be required for conversion of milk to curd. Saccharomyces cerevisiae, that is the baker's yeast. Okay, it will be required for the bread or ethanol formation. Propionibacterium ceremani, it is required for the Swiss cheese. Okay, जिसकी वजह से the large holes are created of the carbon dioxide gas in the cheese. Then Aspergillus niger, it is again a yeast. Okay, and it is required for the formation of citric acid artificially. Now comes the next question. Section C. That will be the 3 mark each question. Eleventh number of question. Describe the process of parturition in the human. So how the process of parturition, or we can say that how the process of delivery takes place in the human female. Okay, that is signals originate from the fetus and the placenta. They both are responsible for the generation of signals. It induces uterine contraction. वो क्या करता है, यूटेराइन के अंदर कॉन्ट्रैक्शन क्रिएट करता है. Then because of this contraction, the oxytocin will be released by the pituitary gland. Oxytocin acts on more on the uterine muscles and it creates stronger uterine contraction. और ज़्यादा uterine contractions create होते हैं. Then uterine contraction and oxytocin secretion continues, resulting into more and more, and after that the baby will be expelled out of the uterus. Now comes the question number 12. Describe the development of endosperm after double fertilization. In an angiosperm, why does endosperm development precede that of the zygote? ठीक है, तो एंडोस्पर्म का डेवलपमेंट कैसे होता है? एंडोस्पर्म that is formed with the help of secondary nucleus, है ना. प्राइमरी एंडोस्पर्म न्यूक्लियस कैसे बनता है? Secondary nucleus plus one male gamete. They both unite with each other and it forms the endosperm. Why उसका हमेशा formation पहले क्यों होता है as compared to zygote? Because endosperm in the future it is going to provide the nourishment to the developing embryo. ठीक है, वो nutrition provide करेगा, वो nourishment provide करेगा to the developing embryo. Because developing embryo होगा, it will be required continuous supply of food. That will be given by the endosperm. Now the question number 13. Explain the interpretation of the Charles Darwin. Okay, this question is omitted as this is from the question chapter number 7, that is evolution. Question number 14. A teacher wants his or her students to find the genotype of a pea plant bearing the purple colored flower in their school garden. Name and explain the cross that it will be possible. Okay, so the same question was there in the 2016. So you can see that 2016 में भी question आया, 2015 में भी ये आया, that is repeated question, and in 2016 also that was 3 or 4 mark question. But इसके अंदर you can show it with the help of a cross. Purple flower to be crossed with 5 white flower. Okay, all the flowers of the urban generation will be homozygous dominant, that is all the flowers will be purple colored. If 50% purple and 50% white, then the genotype is a heterogametic. So this you can show it with the help of a test cross. आप बना के दिखा सकते हो. Question number 15. A DNA segment has a total of 2000 nucleotides, out of which 520 are the adenine containing nucleotides. Total 2000 nucleotides में से 520 are the adenine. How many purine bases this DNA segment possesses? So draw the diagrammatic sketch of a portion of DNA segment to support your answer. Okay, fine. So there will be 1000 purine bases. Okay, because how many purine bases? कितने होंगे purine bases? हाफ होंगे, that is 1000. Now let's see the calculation of it. क्या दिया हुआ है? Adenine 520 दिया हुआ है, and we all know that adenine it always pairs with thymine. तो अगर adenine 520 दिया हुआ है, thymine will also be the 520. Adenine thymine they sum up to form 1040. अब G plus C क्या होगा? That will be 2000 minus, because G plus C कितना होगा? 2000 में से उतना पोर्शन माइनस कर दो, that is 960. अब जितना C होगा उतना G होगा, that's why we are going to divide it by 2. 960 divided by 2, that is 480. Okay. Now pyrimidine आप देख सकते हो कि there are two types of pyrimidine, cytosine and thymine. Okay, plus uracil can also come here if it is asked for the RNA. और purines कितने होते हैं? Purines they are the adenine and guanine. Okay, तो आप कैलकुलेशन करो. C कितना है? 480. थायमिन कितना था? 520, that is 1000. pyrimidine अगर 1000 हो गया तो purine will also be a 1000. Then and only then it can sum up to the 2000. Okay, and we can also solve it with the help of the Chargaff rule. And the Chargaff rule we all know very well, that is A plus T by G plus C will always be equal to the 1, or A by G equal to T plus C will be equal to 1. So this you have to draw it by yourself, okay, to show the base pairing. And when we are drawing the diagram, we have to show the polarity, that is 5' to 3' and 3' to 5'. Nitrogen bases we have to show, hydrogen bonds we have to show, and we have to also show the deoxyribose sugar. These are the four main things that you have to show. Then and only then for the diagram you are going to have the 2 marks. Now comes the next question, that is question number 16. Recombinant DNA technology is of great importance in the field of medicine. With the help of flowchart, show how this technology has been preparing genetically engineered human insulin. So we have to show it over here, that how we can prepare the genetically engineered insulin. Okay. Hello Kushnit. Good afternoon. So let's see. So if it is asked for the flow chart, then we have to draw that only. So we all know first that insulin, ठीक है, human body का जो insulin होता है, it is having the 2 chains, that is A chain and the B chain. Okay, और वो दोनों chains जो होती है, they are joined with the disulfide bond. So when we are preparing this insulin in our laboratory with genetically engineered, okay, तो हमें क्या करना है, ऑर्गेनिज्म इकोलाई लेना है, उसके प्लाज्मिड के अंदर ए चेन वाले जीन को इंसर्ट करना है, दूसरा इकोलाई लेना है, उसके अंदर बी चेन वाले जीन को इंसर्ट करना है, उससे उसकी कॉपीज बन जाएगी, तो एक्सट्रैक्ट किया, ठीक है, एंड देन वी हैव टू जॉइन इट विद द डाइसल्फाइड बॉन्ड, ओके, एंड दिस वाज डन फर्स्ट बाय द एली लिली कंपनी, don't forget that. Now comes the question number 17. Draw the labeled sketch of this staggered stirred tank bioreactor. Staggered stirred. Okay. Write it application also. As it is asked for the stir, when we are making this bioreactor, we have to put two stirs, and it is staggered. Okay, it means it will be having the continuous supply of air. So there will be नीचे की साइड क्या हो जाएंगे, there will be small holes from जिसके नीचे से there will be the passage of air. Okay, air bubbles होंगे, cultural growth होगा, and we have to write the application, right? Correct diagram 1 mark मिलेगा. Any two correct labeling, मतलब कोई भी दो correct labeling कर दोगे, you will be having half mark. हेलो प्रणीता, हाउ आर यू, गुड आफ्टरनून. एप्लीकेशन देखते हैं. प्रोड्यूसेस लार्ज बायोमास लीडिंग टू हायर यील्ड ऑफ डिजायर्ड प्रोटीन्स, recombinant proteins, conversion of raw material into specific product, and there are many other applications also. Then comes the question number 18. Following the collision of two trains, a large number of passengers are killed. Okay, a majority of them are beyond recognition. Authorities want to hand over the dead bodies to their relatives. Name the modern method and write the procedure that it will help for the identification of friendship. So let's see what can we do. We can go for the procedure of DNA fingerprinting. Okay, so if anyone will say that this that person is my relative, so we can the scientist can go for the DNA fingerprinting method. मैम आप HSC बोर्ड का भी समझा रहे हो? Yes, this is for the HSC board. क्यों? HSC board. अच्छा, no. You are talking about your board, na? No, this is the CBSE board itself. But the questions will be almost same because PDF forward करती हूँ मैम, मैं आपको PDF forward करती हूँ. Okay, no problem. You can forward me when I will come on the Unacademy. Okay. And even I am also there on the Telegram app. You can join me over there if you want to ask any doubt or any query. What is the procedure? Isolation and digestion of DNA by restriction endonuclease. Then there will be the separation of DNA fragments. Hybridization using VNTR probe. Detection of hybrid DNA fragment by autoradiography. Matching the bending pattern between the reference DNA and the DNA of the that person. So we can, if they are matching, then we can have the confirmation that this person is their relative or not. Okay. Now comes the question number 19. A team of students are preparing to participate in the inter-school sports meet. During a practice session, you find some vials with label of certain cannabinoids. Okay, fine. Will you report to the authorities? Why? Okay. So what happens over there that you were preparing for the inter-school sports meet, and you have find some vials, and that vials were labeled, vials means small cubes. they were labeled with cannabinoids. Okay, so are you going to report to the authorities? Yes, of course you should report. Because why? Because it is a steroid. Because it may be abused by the sports person. Name a plant from which the chemicals are obtained. So it is the cannabis plant. Okay, from cannabis plant it is obtained. Write the effect of this chemical on the human body. Human body पे इसका effect क्या आएगा? It will affect the cardiovascular system. Okay, it is going to affect the cardiovascular system of the person who will consume it. Now comes the question number 20. Many plants and the animal species are on the verge of extinction because of the loss of the forest land by indiscriminate use of the humans. As a biology student, what method would you suggest along with its advantage that can protect such threatened species from getting extinct? Okay, so you are understanding this question very well, because this is the very common thing that happens. और आपको इन्फॉर्म भी किया है. Yes, मैम ज्वाइन और आपको इन्फॉर्म भी किया. Okay, okay, so I have forwarded maybe. A minute. Yes, yes, yes. I knew it that you have joined it. Sorry. So as a biologist, what method you can give? You can give the conservation method as a ex situ conservation, because threatened animals and the plants are taken out of the natural habitat and placed in a special setting where they are protected and given special care. Okay, so we can go for the ex situ conservation, where we can take these animals, endangered animals, take away these endangered animals from that particular habitat, and हम उनको एक अच्छी ऐसी जगह रख सकते हैं जहाँ पे हम उनकी प्रोटेक्शन कर सकें, एंड वी कैन गिव देम द स्पेशल केयर. Others are the we can prepare the biological gardens or tissue cultures, micropropagation seed culture. Okay, then the optional question: determination of biological oxygen demand can help in suggesting the quality of the water body. Okay, so why it is advised that whenever you are consuming any water, okay, और whenever कभी भी आप कोई भी इंडस्ट्रियल वाटर जब आउटसोर्स हो रहा है, तो हमें उसका बीओडी चेक करना है. क्यों ऐसा एडवाइस किया जाता है? बिकॉज अगर बीओडी बहुत ज़्यादा हुआ, इट मीन्स वाटर की क्वालिटी बहुत ज़्यादा ख़राब है, एंड इफ बीओडी कम हुआ, then we can say that the water is kindly good as per its quality. ठीक है, बिकॉज बायोलॉजिकल ऑक्सीजन डिमांड जब बहुत ज़्यादा है, इट मीन्स वाटर के अंदर ऑर्गेनिक मैटर बहुत ज़्यादा है, और उसके अंदर हम बोल सकते हैं कि ऑर्गेनिक मैटर बहुत ज़्यादा होने की वजह से माइक्रो ऑर्गेनिजम्स का नंबर बहुत ज़्यादा होगा. और अगर बीओडी कम है, ठीक है, तो इस केस में हम क्या बोल सकते हैं कि वाटर के अंदर ऑर्गेनिक मैटर बहुत कम है. ऑर्गेनिक मैटर कम होने की वजह से the number of microorganisms will be less and the quality of water will be good. Clear? So as I have already told you that BOD is a related question, they are having the high chances to be get asked in your examination. Okay, so kindly note it and go through that question again. Now comes the question number 21, and list the steps involved in breeding of a cattle. Suggest any two disadvantages of its practice. Fine. Inbreeding involves mating of closely related individuals within the same breed for 4 to 6 generations. तो inbreeding हम क्या करते हैं? We are mating the organisms within the same breed for around 4 to 6 generations. Superior males and superior females are identified and mated in pairs. ठीक है, progeny को जब हम evolve करते हैं, evaluation करते हैं उसका, तो superior male and female among the selected for further mating. बट इसके अंदर डिसएडवांटेज बहुत बड़ा होता है. मैंने 2 ही डिसएडवांटेजेस पूछे बट बहुत बड़े होते हैं. द डिसएडवांटेजेस इज द मोस्ट इम्पॉर्टेंट इज द इनब्रीडिंग डिप्रेशन. ठीक है, तो इसमें क्या हो जाता है? देयर अकर्स द इनब्रीडिंग डिप्रेशन. वही ऑर्गेनिज्म दे आर अगेन डूइंग द ब्रीडिंग, अगेन डूइंग द ब्रीडिंग, अगेन डूइंग द ब्रीडिंग. ठीक है, तो अगर कुछ भी म्यूटेशन वगैरह कुछ होगा तो इट विल बी रिपीटेड जनरेशन आफ्टर जनरेशन. कोई भी टाइप का म्यूटेशन अगर हो गया तो दैट विल बी रिपीटेड जनरेशन आफ्टर जनरेशन, एंड उसकी वजह से क्या हो जाएगा? इन ब्रीडिंग डिप्रेशन कॉज हो जाएगा. रिडक्शन इन फर्टिलिटी, दे आर हैविंग हाई चांसेस, एंड रिडक्शन इन प्रोडक्टिविटी. Now comes the question number 22. Choose any 3 microbes from the following which are suited for the organic farming, which is of great demand these days for various reasons. Mention one application of one chooser, of each one chooser. Okay, so the organisms are mycorrhiza, monascus, anabina, rhizobium, methanobacterium, and trichoderma. So I will be choosing the most common, and you people also like those microorganisms that are the mycorrhiza, anabina, rhizobium. mycorrhiza का काम क्या होता है? जब हम phosphorus के बारे में पढ़ रहे थे, in that we have only learned about the organism was the mycorrhiza, that is having the high capacity to absorb the phosphorus from the soil. Anabina, it fixes the atmospheric nitrogen, adds organic matter, and rhizobium also, it fixes the atmospheric nitrogen in the leguminous plant. Okay, when you are talking about the rhizobium, don't forget to mention the word leguminous plants. Now comes the section D, that will be of the 4 mark each question, and it is only having the one question. Okay, and this question will be the thoughts or the critical based question. Now comes the question number 23. Since October 2, 2014, Swachh Bharat Abhiyan has been launched in our country. Write the views to initiate giving justification. As a biologist, give any 2 problems that you may face by implementing the program in your locality. Suggest 2 remedial methods to overcome these problems. Okay. So why it was created? Because to give the value to the clean environment and the surrounding. Okay. And being a biologist, 2 problems that can be faced will be सबसे important problem होगी lack of awareness, because people itself are not at all aware about the cleaning. Okay, so if you will take your first step, the people will think that you are mad enough that you are doing the process of cleaning. Okay, that is the first problem that you are going to face. Then social attitudes will be there. Okay, separation of biodegradable and non biodegradable waste, वो भी बहुत बड़ा क्वेश्चन होगा, right? What can you do? You can go for the camping, creating awareness, okay, organizing competitions. Then you will prepare जैसे अभी मास्क ग्लव्स, इनका तो कितना ट्रेंड चल रहा है. 2015 से ही उनको पता था कि they we all are going to use the mask. Okay. Now comes the section E, that is the 5 mark each question. Explain the genetic basis for the blood grouping in the human population. Okay, so one of my favorite questions. I like to answer to this such type of question. And again, it's a very important question, dear students. So okay, so when we are writing about the blood group, okay, so there comes the gene, that is I. If it is IA type, then the blood group will be A blood group, IA or IA and small I. तो क्या हो जाएगा, blood group will be A. IB, so same condition happens in that also, that is IB or IB small I, then it will be the blood group B. For AB type of blood group, the genetic basis should be IA and IB, and for the O there is only one, that is small I and small I. That is, small I is the recessive gene, we can say, that is blood group O occurs only in the recessive gene characters. ठीक है, तो जब तक स्मॉल आई और स्मॉल आई दोनों साथ में नहीं आएंगे, दैट इज दोनों रिसेसिव जीन जब तक साथ में नहीं आएंगे, द ब्लड ग्रुप ओ कैन नॉट बी ऑब्टेन्ड. अंडरस्टूड? Now the third point, since humans are diploid, each person possesses any two of the three alleles. ठीक है, जैसे हमें पता है कि ह्यूमन बीइंग का जो जीन है दैट इज डिप्लॉयड इन नेचर. ठीक है, तो कोई ना कोई दो एलिल्स तो आएंगे ही आई के, रिजल्टिंग इनटू 6 डिफरेंट जीनोटाइपिक कॉम्बिनेशंस एंड 4 फिनोटाइपिक कॉम्बिनेशन. तो जीनोटाइपिक तो 6 थे, 1 2 3 4 and this is 5 and 6. Okay, there were just 6 different genotypes and 4 phenotypes. Got it? Now comes the next question, again a very important question, and as I have already told you, the most important and the repeated question. How did Hershey and Chase establish that the DNA is transferred from virus to bacteria? How they have informed? Okay, तो उसके अंदर आएगा हर्शी एंड चेस का एक्सपेरिमेंट, जिसके अंदर उन्होंने P32 और S35 वाला रेडियो एक्टिव फॉर्म यूज़ किया था. ठीक है, P32 they have used for the radioactive implantation in the virus DNA, और S35 उन्होंने यूज़ किया था ताकि उसकी प्रोटीन कोड को वो रेडियो एक्टिव फॉर्म में कन्वर्ट कर सके. देन दे हैव इंजेक्टेड इनटू द इकोलाई. ठीक है, उसके अंदर इन्फेक्शन किया इकोलाई के अंदर, सेंट्रीफिकेशन किया, दे हैव फाउंड दैट जब प्रोटीन ट्रांसफ़र हुआ था तो इकोलाई के अंदर रेडियो एक्टिव सल्फर एब्सेंट था. ठीक है, व्हेन देयर वाज अ ट्रांसफ़र ऑफ प्रोटीन, ठीक है, तो उस टाइम पे इकोलाई के अंदर radioactive form of sulfur was absent, but when there was transfer of a DNA, okay, at that moment in the E. coli there was the presence of phosphorus, that is radioactive phosphorus उसमें present था. So with the help of this experiment, the Hershey and Chase were able to explain that the DNA is getting transferred from one generation to the another generation, or when there is an infection of one organism to the another. We can show it with the help of experiment also, diagram also. This diagram you all know very well that it is given in your textbook, right? So you have to draw the diagram, as it is the 5-mark question, so you cannot just write the points, answer in theoretical form and you can finish. You have to mention the diagram also, because diagram is also having its own weightage. You can see diagram weightage. Okay, now comes the question number 25. Analysis of age pyramid for the human population can provide important input for long-term planning strategy. Explain how. Okay, age pyramid, it shows the age distribution of male and female combined in the combined diagram. एक ही diagram के अंदर by making the age pyramid we can come to know about the male and the female distribution of ages. Okay, The shape of pyramid reflect the growth status of the population, where it is growing or stable or decline. तो by looking at the pyramid, हमें pyramid से ही पता चल जाएगा कि pyramid इस टाइप का है कि इस टाइप का है या अजीबोगरीब है, कुछ भी शेप है पिरामिड का. So by looking at the pyramid, we can come to know whether the growing population it is stable or it is declining. Stably move हो रही है या वो decline हो रही है. Pyramid also indicate the ratio of pre-reproductive, reproductive and post-reproductive individuals in the population. So by looking at the pyramid, if the number of young ones, okay, newborns will be more and the number of old person will be very less, तो हम इस टाइम पे क्या बोलेंगे, प्री रिप्रोडक्टिव पॉपुलेशन इज मोर एज कंपेयर टू दी पोस्ट रिप्रोडक्टिव. ओके, हेलो सांद्रा सी जी, so you are bit late, no problem, but you didn't knew that my lecture was at 2 p.m. or you were busy at some other place. Okay, और अदर पॉपुलेशन के अंदर प्री रिप्रोडक्टिव मतलब न्यू बॉर्न बेबी ऑलमोस्ट दे वर इन द लेवल, देन द रिप्रोडक्टिव पीपल्स वेर मोर एंड द पोस्ट रिप्रोडक्टिव लेस. ठीक है, तो उस टाइप की पिरामिड को शेप कैसा होगा, इट विल बी लाइक, ओके. सो यस, हियर इज द डायग्राम. एक्सपांडिंग दैट इज प्री रिप्रोडक्टिव वाले ज़्यादा है, रिप्रोडक्टिव वाले उनसे थोड़े कम है, पोस्ट रिप्रोडक्टिव बहुत कम है. मतलब एक्सपांडिंग पॉपुलेशन की पॉपुलेशन इज इंक्रीजिंग अलॉट. टेबल में क्या हो जाएगा, प्री रिप्रोडक्टिव और रिप्रोडक्टिव दैट इज न्यू बॉर्न बेबीज एंड दी यंग वंस दे आर ऑलमोस्ट इक्वल. ठीक है, एज कंपेयर टू दी पोस्ट रिप्रोडक्टिव. डिक्लाइनिंग में प्री रिप्रोडक्टिव बहुत कम, reproductive stage that is the young ones they are quite more as compared to the pre-reproductive and the post-reproductive. So such type of diagram it will show the declining population. So what can we do? We can plan for the health, education, transport, infrastructure, finance, food, employment, anything. Now describe the advantages for keeping the ecosystem healthy. It's a very general question. Is it so? So it is purifying the air. How we can keep the ecosystem healthy? Okay, purify air, purify water, mitigate the drought, cycle nutrients, generate fertile soil, provide wildlife habitat, maintain biodiversity, pollinate crops, provide storage, fight for the carbon, and there are many other advantages and disadvantages. Now comes the question number 26. Is it visible to you, right, students? A flower of tomato plant following the process of sexual reproduction produces 200 viable seeds. Okay, so a flower of tomato plants which is having the following the sexual reproduction, it is going to produce 200 viable seeds. To give the reason, what would have been the minimum number of ovules present in the pre-pollinated pistil? So this question is a very boring type of a question, but it is a critical based question. So let's see. First of all, we will read the question again. A tomato plant following the process of sexual reproduction produces 200 viable seeds. ठीक है, एक टोमेटो प्लांट था जिसने सेक्सुअल रिप्रोडक्शन किया and it produces 200 viable seeds. We have to answer the different questions. What would have been the minimum number of ovules present in the pre-pollinated pistil? तो कितने होंगे ovules? अगर 200 viable seeds बना रहे तो ovules will be 200 itself. जितने ovules उतने seeds बनाएगा, है ना? Then how many microscope mother cell could would minimally be required to produce the requested number of pollen grains. तो कितने हमें चाहिए होंगे? तो अगर 200 ओव्यूल है, ठीक है, तो 200 pollen grain भी चाहिए होंगे. And to produce the 200 pollen grains, we at least require 50 microspore mother cell. कितने चाहिए? 50, because this 50 will have undergo the process of meiosis. वो क्या करेगा? meiosis करेंगे 50 cells. So they are going to produce the 200. Okay. Then the C number of question. How many pollen grain must have minimally pollinated the carpel? कितने pollen grain से pollination किया होगा? 200 ने तो किया ही होगा. Okay. Then how many male gametes would have used to produce these 200 मेल वाइबल सीड्स? कितने मेल गैमेट्स चाहिए होंगे? तो मेल गैमेट्स चाहिए होंगे, दैट विल बी दी 400. ठीक है, बिकॉज मेल गैमेट्स दे ईच पॉलन ग्रेन हैज 2 मेल गैमेट्स, है ना. एक पॉलन ग्रेन में 2 मेल गैमेट्स होते हैं. ठीक है, तो मेल गैमेट्स कितने होंगे? 400. MMC दैट इज माइक्रोस्पोर मदर सेल विल बी 200. सबके अंदर एक एक फंक्शन हो जाते ही बाद में. Now the optional question. Explain the development of secondary oocyte in the human female from the embryonic stage up to its ovulation. Okay. From the embryonic stage till the ovulation we have to explain. Name the hormones involved in this process. Okay. So now you can see over here fetal stage, है ना. So when the female girl child, okay, when the girl child is born, she is always born with the primary oocytes. उसकी बॉडी में प्राइमरी oocytes तो होते ही हैं, that is oogonia. It has undergo the mitosis process. Primary oocytes बनाया. Now when there will be the I can say that is वो first meiotic division कर लेता है. Primary oocyte it undergoes the process of first meiotic division when there is a child born. ओके, फिर बाकी का फर्स्ट मियोटिक डिवीजन के बाद सेकेंड मियोटिक डिवीजन करना या फर्स्ट मियोटिक डिवीजन को भी कंप्लीट करके आगे बढ़ना. सो दैट अकर्स एट द प्यूबर्टी, दैट इज एज इट इज शी इज गोइंग टू रीच हर एट द एज ऑफ 12 to 13 आई कैन से, और 11 to 13. ठीक है, एट दैट मोमेंट इट इज गोइंग टू प्रोड्यूस द सेकेंडरी oocyte. She is going to prepare the secondary oocyte and the first molar body. Now the ovulation occurs at this stage. इस स्टेज पे क्या हो जाता है? ओव्यूलेशन हो जाता है. दैट इज सेकेंडरी oocyte is released, okay, with the first polar body, and if the fertilization will occur, then secondary oocyte is again going to go into the meiotic division and will form the egg cell. Diksha Kashyap, hello Diksha. Afternoon. गुड आफ्टरनून रितांशु. अभी हमारी 1 मिनट में क्लास एंड होने वाली है. एंड यू आर सो लेट माय डियर. व्हाई इज इट सो? So the hormones which are required, they are the LH, that is luteinizing hormone, FSH, follicle stimulating hormone, और estrogen. Okay, but बच्चा बड़ी एक चीज ध्यान रखना, when you are writing the exam, okay, सब फुल फॉर्म भी लीक हो सकती है. ओके, काफ़ी सारे बच्चे ऐसे होते हैं जो शॉर्ट फॉर्म लिख के जाते हैं, FSH, LH. मैम फुल फॉर्म तो आपको ही पता होगा ना. ओके, so don't do that mistake. Always mention the full form with the short form. Okay, so it gives good impression of yours. So that was all about the paper. Okay, so my dear bachcha body, thank you so much for attending my today's lecture. I know many students have joined it right now, they are so late, but as the time is completed, I have to end my session. Don't forget to make use of the code, that is DG007, that for having the 10% discount in your Unacademy subscription. We are going to meet again at 9 PM today on the Unacademy application. In that we are going to have our discussion on the 2016 question paper. Okay, on the Unacademy application. Till then, कल तो संडे है, कल तो हमारी यूट्यूब पे छुट्टी है. मंडे को हम मिलेंगे on our same timing, that is 11 a.m. in the morning and 2 p.m. in the afternoon. Sorry ma'am, I'm late. No, nothing to say sorry, Dipanshu. It's completely fine. Okay. Chalo, bye bye, bacha buddy. Don't forget to make use of the referral code DG007. Okay, if you want Divya ma'am to teach on the Unacademy for the long period of time. Okay, till then, bye bye. Take care, everyone.